Hello, I'm very pleased to be with you virtually today to discuss a talk about an evolutionary theory of aging. And more specifically, today, I will tell you about age-distorters, which I believe are entities able to affect aging of an host from a different species. I am an evolutionary biologist, and I study evolution using networks. So this is what I really do for a living. I try to understand where the diversity of traits and the diversity of species comes from. But today, I will have a specific focus on the evolution of aging. And that is a question for the evolutionary theories, which tries to explain how and why we age. This is very debated, and I guess there are a few final answers to that question right now. The plan of my talk today will first consist in showing you that there is a problem, at least one, and there are many, in fact, but there is one with the evolutionary theories of aging because they admit one blind spot. So they are missing something about aging. And the things that they are missing, I believe, are the age-distorters. So I will introduce you what age-distorters are, these entities that manipulate the aging of one host for their own benefit. And I will give you then evidence that there are age-distorters known in the literature and that they probably are around us. Then I will focus on the scientific importance of age-distorters. Why does this matter to give a name to this entity that may manipulate the aging of other entity? And then I will ask ourselves a question, what is next? What if we find these age-distorters? Is it going to be a good news and why? So I want to start with a simple kind of explanation about what evolutionary theories of aging do for us. What they do is that they are inspired from the evolutionary theory in general, and this evolutionary theory, as you know, is strongly impacted by population genetics, which is a very good science, very important, and basically tells us how within a population, and therefore within a species, various traits came to be and be selected for. So in population genetics, you care for what happens to the genes of your population, to the genes of your species. And logically, because evolutionary theory of aging come from population genetics in a sense, they also stress the genetic components that are important for aging that comes from one species. For example, the most classic, in the sense most ancient evolutionary theory of aging is called the mutation accumulation theory. It was proposed by Medawar. It dates from 1952, and it is very interesting in the sense that it explains aging as a consequence of the accumulation of deleterious late expressed genes, genes that are expressed late in life and harm us. And there is a good evolutionary reason why, indeed, it may be the case for a long-lived species that this species could have bad genes expressed late in the life of the organism. And the reason is like this: as you live longer, you face more and more risk to die of accident. And therefore, the probability that you are still around when you are older to reproduce decreases with time, just because you are going to possibly have an accident that will kill you, and the longer you have been around, the higher the risk. So the probability that you're dead is also higher when you're old. Low probability of reproduction and high probability of death mean that the contribution of old individual is going to be limited for the next generation. And if they have some good genes that are keeping them in good shape when they are old, it is unlikely that their offspring will live as old as the parents, because the offspring would have to survive quite a few accidents. And therefore, in the conventional theory, what we have in evolution is the notion that when natural selection is active, which is basically from the time you are born to where you are at your peak of sexual maturity, then natural selection is going to sort of purge your bad genes from your genome. But later on, as the strength of natural selection decreases with time, then some terrible genes may be expressed and harm you. But natural selection has no way to counteract that because it is too weak and it is too late, in a sense. And so typically, Huntington's disease has been proposed to be one of these terrible disease where late expressed genes expressed after the age of sexual maturity is not going to be removed from the population because there is not a strong enough selection to remove it. And so that's the theory. It says you have some genes that are bad for you when they're expressed later, and natural selection cannot save you. Another theory that is also very classic is called Antagonistic pleiotropic theory. It dates from 1957. It has been proposed by Williams. And what it tells us is that indeed, selection is stronger when you are younger, and it doesn't foresee the future. So if there is something that do you good when you're young, selection is likely going to favor it. Typically, a gene that will enhance your chances of reproducing is going to be favored, even if later on the expression of the same gene is going to be costly and have negative deleterious effect on your life. So a gene that would be good for your reproduction early is going to be favored, even if it's bad for your survival later. And for example, for a man having high testosterone level may be a chance to be more active, seductive, and whatever you want when he's young, but then later it may also raise the risk of cancer prostate. Again, one explanation of aging that has to do with the genes within a species. And finally, there is another major proposal for explaining aging in animals, and that is a disposable soma theory proposed by Kirkwood in 1977. And what this theory tells us is that animals are, in many cases, nicely compartmented in two parts: the soma, that is the body supporting most of the living functions, and the germline, that is a part of your body that will basically propagate the lineage from which your next generation will come off. And what the disposable soma theory suggests about the evolution of aging is that an organism will age when basically it uses its energy, it uses its resources, it invests it to preserve the germline and enhance the chances to have a future generation at the cost of the maintenance of the soma. So this is a balance in a sense. And if your energy goes into your growth and reproduction, then it won't go to fixing your body and you will age and you will decay. And eventually you will do this very fast in the case of the salmon. So all these theories are especially important because they agree on one point: aging is a byproduct. So there is no gene that are selected to get you to age. In the case of mutation accumulation, you are aging because basically evolution is letting you down, natural selection is not strong enough, so it's an evolution neglect. In the case of antagonistic pleiotropy, you are aging because your reproduction is optimized. That's what you're aging. And in the case of the disposable soma theory, you're also aging because there is an energy allocation for reproduction that is optimized. But again, aging is a consequence of that. And importantly, in all these three theories, you notice that the focus is on individual within one species. However, we now know that the life cycles of many organisms intersect. And so this is my first point. The common blind spot for all these theories is that they overlook the fact that humans, for example, are not living alone. They are infected by viruses, by bacteria, they have microbiomes. And so because all of this life cycle intersect, one may wonder whether this intersection may have an impact on the evolution of aging. But currently, this is not modeled in the theory. So what if aging in one species involves the genes of another species? Here's my question today. So introducing the notion of age-distorter more precisely, age-distorter would be these entities that are able to manipulate the aging of one host for their own benefit using their own genes. Why do we believe such a thing could work? Well, there could be one type of age-distorter that would basically carry genes, age-distorting genes, that would allow the age-distorter to interfere directly with the genes that are involved in the aging of the host. So here you have in black drawing of a pathway with black nodes that are involved in getting an host to age. And this is because within the species, there have been many terrible genes that are there, or genes that are allow for the allocation of more energy to the germline than to the soma, or maybe those are your antagonistic pleiotropic genes. But at any rate, what we have in addition on this drawing are the red nodes, and the red nodes correspond to genes carried by the age-distorter. And what are those genes doing? They either regulate the host genes that are involved in aging, or they mimic the host genes. They could even be copies of the host genes, but not carried on the genomes of the host, now carrying in the DNA of the age-distorter. And there is a second type of age distortion that is very possible. And it goes like this: not involved directly with an interference with the host aging pathways, an age distorter can carry genes that result in more damages to the host. So if you have this situation and you have an age distorter that brings in your body more genes than do you bad, it accumulates the damages, and therefore you will need more energy to self-maintain. And eventually, if you don't have more energy, you will age because the damage load is stronger. So age distorter, I will argue, could affect the aging of entire organism. So they could basically lead an organism faster or slower through its life cycle. And eventually, if it leads to a faster senescence, we will observe in nature senescent organisms because they are infected by age distorter, or it could affect part of the organism, and in which case, excuse me, it would affect the cell cycle, and it may result in quiescent or senescent cells if, for example, the age distorter needs to replicate inside a senescent cell for its own good. So do age distorters exist? Do we have any evidence that those things are real? I would argue that yes, and that viruses are first a very good candidate for this, that pathogenic bacteria are second a good candidate for age distorters, parasites in general, symbionts in general, that basically share a host body, and when they use the host body, in some sense, appropriate themselves part of the host body. And also the microbiome is very important because, as you know, human beings carry a microbiome, and only in their gut there are many more genes from microbial origins, 150 times more or 200 times more genes that are from the microbiome than from our own genome. Human genome is associated with our body. So why viruses first? First, as you know, viral infections are very common. They can be widely distributed. They are also easily transmitted, and it is challenging to control them. But also, you know that viruses, for their own life cycle, they need to go inside one host cell, and they need to take control of the cellular machinery to replicate. So at some point, there is something in the biology that is taking place, and that thing is the virus takes the leadership inside the cell that is infected, and the virus tells what genes are going to be expressed, and for its own good. It doesn't care for the cell anymore at that stage. An example of a viral gene that is affecting an entire behavior of an organism can be seen in an old paper from Hoover. It's a very nice paper, by the way. And this is a gene from viruses that induces a disease in the caterpillar. And the caterpillar, when infected by the virus, will climb, and then it will die and melt. And a rain of viruses will fall over non-infected caterpillar that then will also have this strange behavior. So it is admitted and known that viruses can manipulate cells and that they can manipulate entire organisms. Now, not everybody is familiar with that notion, and most of us know that when viruses attack a cell, basically they steal the resources from the cell for their own benefit, and then the virus multiply, and they're happy to do this. But there is another strategy for viruses, and that strategy is basically to take control for a longer time of the host cells, or of the host organism, to their own benefit. And there is one case, I will argue, when the viruses take control in a way that leads the host that is being controlled, of which a machinery is hijacked, to age. So do we have an example of viruses that are aging their host? Yes. And HIV-1 is probably a good example of this. As you may have read in the literature, an infection by HIV-1 may accelerate even your epigenetic clock, meaning that infected cells are aging faster, and infected organism, infected hosts, infected humans are aging faster. And in particular, there is this process of immunosenescence that is terrible, and that leads to a less effective immune system to fight against the virus. So you can see that there is an evolutionary interest for the virus to get our immune system older, and to get us older because it helps the virus to better propagate. And the same is true for some viruses that are inducing senescent cells. For example, the human papillomavirus or the varicella zoster virus are basically happy when there are senescent cells in our body, and senescent cells, as you surely know, are no longer able to replicate, but they are still efficient in terms of metabolism, and they do something that is problematic when you are old, especially to the neighboring cells. The senescent cell is sending around SASP, which are various molecules that are inducing a senescent phenotype in the other cells nearby. So you start with a senescent cell, and the senescence propagates in a bystander effect in some systemic way, and you end up having an aging tissular environment. So when you have a virus that basically generates a senescent cell, then it has, by bystander effect, a larger effect on the host, because then there will be more and more senescent cells, and eventually that will accelerate aging in one organism. We have example of bacteria playing a similar role, not only viruses, and this is, for example, the case of Salmonella typhi, which is in red here, enters inside a host cell, a human host cell, secretes some typhoid toxin, and the typhoid toxin is going to enter inside human nucleus and generate DNA damage. So DNA damage is terrible in many respects in that case, because it can be fixed when there is not enough energy to fix the DNA damage. And eventually, too much damage means that the repair machinery of the host will be exhausted. And when it is exhausted, damage accumulates. And when damage accumulates, the cells that is infected become senescent. And when you know that the cell is senescent, then the senescence propagates nearby. And so you have aging by bystander effect due to this bacteria that is distorting the age of the infected cells and its vicinity. We have also examples of microbiomes that are involved in age distortion of the host. This, for example, is the case with the bacteria Coma monas. When it is fed to the nematode C. elegans, then C. elegans will have a shorter lifespan, because C. elegans will actually develop faster, age faster, die younger. And similarly, it has been showed that in E. coli, there are 29 genes that are able to contribute to C. elegans aging. And these genes, interestingly, do this by interacting with some of the host aging pathways, such as the insulin EGF-1 pathway or the TOR signaling pathway, and also some genes that are reducing the ability of the host to repair their damaged mitochondria. So these are, again, more examples of age distorters that you can find through the literature: viruses, bacteria, microbiome. And outside this prokaryotic world, you have also instances of age-distorters. For example, you have animals that may be parasites of other animals and that will behave as age-distorter. In this particular case, I will show you the age-distorter is slowing down aging of the host. It is doing this for a reason. For example, Hymenopletis diminuta is a parasite that infects a beetle, and it will actually get the beetle to live longer because the parasite cannot complete his life cycle inside the beetle. The beetle has to be eaten by rats or other animals for the parasite to complete its cycle. And so if the beetle lives longer, the rats have more chances to eat the beetle and to engulf the parasite. And these kind of discoveries have been made repeatedly over the past 20 years. And this is, for example, a very recent case of a paper where likewise the parasite is expanding the lifespan of its host for its own benefit. So why should one care for age-distorter is the next question, really. I will tell you that they will expand the current evolutionary of aging because this theory had a blind spot and now we are filling in the blind spot. So how will the theory adjust to this? Also because it will show that selection can be involved in the aging of a species, also of another species than the selection acting on the age-distorter, not selection acting on the aging species directly. Also, I will suggest that there may be translational payoff of discovering age-distorters and that this age-distorter could help us better know the aging pathways of the host, including human aging pathways. First, how do this age-distorter enhance our theories? You may remember the classic mutation accumulation theory where I told you with age, rotten genes, bad genes are going to accumulate and they can't be eliminated because natural selection is not strong enough in this selection shadow. What you have is in fact the same for age-distorters if you consider that pathogens attacking you or microbiome living with you, especially when you are old, are under a lesser pressure, selective pressure by the host to be eliminated. And this is because as the host is less and less under natural selection to prevent its bad gene to express, it is also less and less able to prevent the bad genes of its microbiome to express. And so not only rotten genes in our genome will accumulate with time, but also rotten genes in the genomes of our age-distorter. So older people may be more sensitive to age-distorter under that scenario. In the case of antagonistic pleiotropy, remember that the theory explains how a gene in your genome may do you good early, but then later you pay the cost of the expression of that gene. But you also remember that in our microbiome, for example, there are so many more genes that are carried by our microbes. And so if there are some genes in our microbes that actually do us good when we are young, and they may be selected for positively for their presence, then this may be at the expense of a later damaging effect. For example, some viruses are able to induce senescence, I told you, which is good when we are young because it increases, in fact, tissue repairs because the signal from the senescent cell are going to attract somatic maintenance on spot, but having more senescent cells is actually bad when you're already aging because it's too many senescent cells in your body. And likewise, disposable soma theory is going to be expanded. You remember that in this theory, what matters to explain aging is that the soma is abandoned and to the benefit of the germline, and therefore that the energy goes to the growth and reproduction of the germline and not to the somatic maintenance and repair. But if you now consider age-distorters, you have a comparable situation by analogy. You have, in fact, now a given age-distorter in blue that takes control over a part of the host body. And this part may be in the germline or it may be in the soma. The age-distorter doesn't care, but what it will do, it will abandon leftover the part that is under control for the benefit, the reproduction of the age-distorter. And when the age-distorter is actually controlling the entire organism, it will use the entire body and treat it as a disposable soma for its own benefit. So the logic of the disposable soma, if you wish, is preserved by the age-distorter approach, except what becomes a disposable body is now the body of the host. Why does it matter? From a fundamental perspective, because you remember that in the current evolutionary theory of aging, aging will not evolve directly as a result of selection. But here, aging of an host can evolve directly as a result of selection going on another species, when the selection will actually favor the growth and reproduction of another species at the expense of the maintenance, repair, and reproduction of the host. What next? What could we do to better know these age-distorters? I think first we could look for them, try to identify where they are, especially human age-distorters, because they're going to affect our aging. So you remember, age-distorters are characterized by the fact that they encode age-distorting genes. And some of these distorting genes, I told you, are able to interact with the host aging pathways, either because they are the same, homologous, evolutionary conserved copies, stolen copies, if you wish, of the host, and they're present not only in the host, but they're also present in the age-distorter. So when you're infected, you don't have only one copy of the gene, but you have two copies of the gene, and that may have terrible effect on your aging. Or they are regulators of your host aging genes. So one way to find them is basically to look trivially for genes that are present, for example, in viruses and interact with the host aging pathways, or are the same than the one in the host aging pathways. So how do you do this? First step is you can start from protein-protein interaction networks, and this is exactly what you see here. Every node is a protein from either the human aging network or from the cellular aging human network. So those are the genes in white that have been demonstrated to be associated with the aging of human cells or with the aging of human. And in blue, you have the direct protein partners of these white proteins. And so this is an overview, if you wish, of what is known about the proteins that interact with the proteins that are involved in your aging. And what is interesting is that you can now look on that map whether there are some of these proteins that are also present in viruses or in bacteria or in your microbiome. So the way to do this is to perform what is called an homology search. You do that by a computer, a comparison of sequences. And basically, you start from the protein sequences here that are known, and you look for sequences that are very similar in viruses and in bacteria or in your microbiome. And by very similar, I mean here sequences that align over the majority of your protein and that have more than 75% of identity with your protein with an e-value that is not ridiculous. In principle, what could happen when one performs this kind of analysis? Well, you could find first that indeed some of the protein in your usual network are also carried by an age-distorter. And this is represented here in green. And so the green nodes are showing proteins shared by your genome and also genomes of species that interact with your lifestyle. So, for example, in viruses. And if you do that, you can also color the edges going from these proteins in yellow, for example, to show with what other proteins this protein carried by a virus is interacting or is able to interact. When you do that, you can, at the end of your analysis, extract all this interaction and claim that this would be eventually the different part of the pathways involved in cell aging or in human aging that could be manipulated by an age-distorter. For real data on real viruses, this is what you get. And that's only for a subset of the virome that we have investigated. But what you get is beautiful in a sense. You observe here two genes that are present in viruses and are also known to be associated with aging in the human, and 49 partner proteins that are also present encoded by the gene in the viruses, and that are very similar to the human protein partners of the proteins involved in human aging. So you see regions of possible manipulation by viruses. Why would it matter to identify these age distorters? Well, because if we know some of them, we may already have vaccines and we may already have drugs to treat them. If we know some of the genes, age distorted genes that are involved, likewise, there may be drugs that could inhibit the action of these genes. You may also use the notion of age distorter to basically induce as tools, as Trojan horse, more senescence in some of your cells or less senescence in some of your cells, but that has to be done very carefully. And if your human age distorters are in fact in your gut, in your microbiome, then a fecal microbiome transplant may be an option to get rid of these age distorters. So as a conclusion, what I wanted to tell you today is that the evolutionary theory of aging is incomplete when it doesn't look for the co-evolution of aging that is the result of interaction between multiple species, and we know there are interactions between species everywhere. In fact, genetic aging threats could also come from outside our own genome, and if it is the case, then this is a worrying question, but are we individually older than we could be because we made bad encounters and because we have our aging manipulated by other species? And with that, I would like to conclude and thank very much everybody who is in charge of the analysis in my lab, Jérôme Tellier, Debashish Bhattacharya, colleague who is working with us on microbial aging, and Philippe Lopez, Eduardo Correll, yourself for your time, and I would welcome new collaboration on the evolution of aging. Thank you very much for your time.